Welcome to Fluidic Conversations. So I'm Victoria. I'm one of the hosts of Fluidic Conversations, where we host discussions on the state of up-and-coming innovative technologies or new discoveries, challenges, and future developments. Following each one of our podcasts, we'll release an issue into our Fluidic Frontiers magazine with our upcoming issue focusing on water in particular. So today we have Dr. Yves Filion, a professor and undergraduate chair in the Department of Civil Engineering at Queen's University. So Dr. Filion is a licensed professional engineer in Ontario, and alongside his research interests, he has experience in consulting for topics ranging from design of drinking water, stormwater, and wastewater systems, as well as infrastructure renewal planning. He's a leading researcher in sustainable water systems and improving drinking water quality in water distribution systems. So just thank you so much for meeting with us today to discuss your research, and we're really excited to learn about it. So if you want, you can give us a quick introduction, and then if you'd like, share your screen and give us a presentation on your research.
Okay, well, I've been a prof at Queen's for about 17 years, and most of my research is focused on looking at different aspects of drinking water in distribution systems. So I started out my career looking at mostly at how we can do, I've shifted to looking at the water quality implications of drinking water, particularly how metals can accumulate in pipes and how they can be mobilized and cause all sorts of water quality problems in the system. So I'll give you maybe a 10-minute overview of what we do here at Queen's in this area. Queen's here, we're the drinking water quality group, so comprised of myself as well as two other researchers, Dr. Sarah Jane Payne and Dr. Cheyenne Zing. And again, we look at different aspects of water quality modeling and water quality transformations in distribution systems. My primary areas of research activity are what we call drinking water discoloration, so how metals accumulate in pipes and how they're mobilized, like I said earlier. I also do some work in energy interactions in distribution systems, so primarily looking at ways to make the system more energy efficient. And we're also, recently we've looked at how biofilms, which are like thin films or bacterial cells that are adhered to the pipe wall, how they can actually contribute to the development of antimicrobial resistance or antibiotic resistance. I did my schooling, all of my degrees, including my PhD at the University of Toronto in the Department of Civil Engineering. And I worked for a time in consulting for this firm called R.V. Anderson Associates. So this is where I come from. I did all of that before joining Queen's University in 2006, where I've been ever since. So looking at discoloration, so this is maybe a profile of a pipe. You have also these weird processes that go on in the pipe. Some of them are hydraulic in nature. Others are chemical in nature, and others are biological in nature. And so if you have particles here in the water, some of them will settle by gravity on the pipe wall at the bottom. Some of it, the flow in the pipe is extraordinarily turbulent, which means that the fluid flow has a very chaotic nature. And these particles can be thrown to the top of the pipe and the sides of the pipe, and they can actually stay there against the action of gravity. Often, drinking water is not perfectly sterile. It has biological material in it, different organisms from bacteria to archaea to protists. And these organisms, of course, want to live like everyone else. And so they tend to stick to pipe and create what are called biofilms. And so they live underneath these biofilms, which are primarily made up of macromolecules like proteins and carbohydrates and lipids and extracellular DNA that actually give physical structure to the film and allow them to sort of be protected from all of the chaos in the fluid right next to the wall. And of course, because you have water flowing next to a solid boundary, which is the wall of the pipe, that induces a mechanical shear at the wall. And so again, these macromolecules that form the biofilm almost act as a cement to keep it in place to resist this wall shear force. So that things don't get ripped away. But unfortunately, sometimes there are extraordinary hydraulic events that go on in the system. Like, for example, when a hydrant is opened suddenly when there's a fire in the system or a pump fails abruptly, that creates rapid changes in flow. And it actually makes accelerations in the flow, which produces these shear stresses that very temporarily are very high. And so they can overcome the strength of this biofilm and actually mobilize not only the biofilm, but also the metals that have adhered to the pipe wall. And that causes what are called red water problems that are sometimes reported in municipalities throughout Canada. They are very disruptive. They cause customer complaints. They ruin people's wash because red iron oxide particles get into the washing machine, and they cause red staining on the clothing. And they can also be very toxic. Sometimes manganese is one of those metals, which has been shown to be a neurotoxin for children. So there's a lot of negative things associated with discoloration events.
Wow, that is something I didn't know. I didn't know that that's what it was caused by, kind of
Yeah.
iron and such. That's actually really interesting,
Yeah.
and I didn't know. Oh, wow. Okay.
So two of the problem metals that show up and that cause problems are iron and manganese. Those are the two. They're the ones too that actually cause a change in color in the water and, tend to anger customers because no one likes to drink red water. So we sort of set out a number of research questions for ourselves here at Queen's. We wanted to know how biofilms grow, sort of mechanisms in place for that, how inorganic particles like iron and manganese actually maybe contribute to growth of biofilms. We're also looking at how the hydrodynamic conditions in the pipe, the hydraulic conditions in the pipe, all the turbulence that goes on, the shear force at the wall, how those can act to not only accumulate or cause particles to adhere to the wall, but how they can be mobilized and cause all of this chaos downstream in terms of water quality. The funny thing is that the accumulation process and the mobilization process take place at very different scales, both spatially and temporally, so in space and time. The accumulation process tends to be microscopic, and so we are very focused on looking at things at the wall with microscopes so we can see how many particles, actually accumulate and adhere to the wall. The mobilization process causes a lot of red water over sometimes kilometers of pipe. So the spatial scale is much, much larger. So that's what makes this research kind of tricky. You have to look at things both at the microscopic scale, like at the micro scale, and even nanometer scale, but also at the meter and kilometer scale. The accumulation process tends to also happen very, very slowly. Sometimes it can take years for biofilm and these metals to accumulate on pipe walls. But if there's a big hydraulic event, like a big pump failure or a big pipe break that causes a big change in flow, all of that material can be released within seconds or minutes. So again, the time scale is extremely disparate when you're looking at accumulation and mobilization. Very tricky scale difference to navigate. So at Queen's, to look at all of these things, we built what's called the Drinking Water Distribution Lab, and there's a photo of it here. It's a large-scale facility with two large-scale distribution systems. So you could see one on the left and one on the right. They're totally separate hydraulically and in terms of water quality. So we can actually run experiments concurrently with this system. And they're full-scale PVC pipes, the same pipes that actually you would encounter in real distribution networks. So all of our research that we do that involves discoloration is done at full scale, which has great advantages because we don't actually have to scale down all of the processes. We just basically run the experiments as we would in a real network, but with a great advantage of having complete control over all of the hydraulic conditions and water quality conditions in the network and the environmental conditions like temperature, something that you don't have when you're working out in the field.
Yeah. And was this made recently, this drinking water distribution laboratory? Is this like,
Yeah, so we were successful with a Canada Foundation for Innovation grant
oh, great.
about 12, 14, 13 years ago.
Oh, okay.
And it took about 5 years to build and
Yeah.
commission. So we've been working with this facility for about 7 years now.
Wow.
Yeah.
That's great.
So we look at all of these accumulation processes and mobilization mobilization processes with this facility. So the facility itself, again, is 2 loops. Each loop has 200 meters of pipe organized in 11 closed loops. And the diameter of the pipe is 100 millimeters, so about 4 inches. It can pass flows between 0 and 16 liters per second, which is quite high. And it's in a temperature controlled room. So basically we run our experiments anywhere between 8 degrees Celsius and 16 degrees Celsius. The graduate students that work in here tend to wear coats when they go into the hydraulic room and actually have to sample, because it's actually kind of a cold chamber. And the reason we run them at those temperatures is because the water in a lot of distribution systems can vary between anywhere from 5 to 8 degrees Celsius to 16 degrees Celsius. So some experiments that we've run, basically the experiments that we run are made up of 2 parts. There's a 30-day, we usually use 30 days to grow the biofilm. And so we introduce nutrients into the water to help the bacteria grow and adhere to the pipe wall. And then after the 30-day period, we inject the system with known concentrations of different metals. So one of the metals that we've been focusing on is iron oxides in particulate form, because that is one of the big culprits that causes discoloration problems. And we do a series of injections to let those particles run through the system, adhere to the biofilm, and settle down. Once that's done, then we flush the system a number of times at different flushing levels, different shear levels, and actually take a series of measurements like turbidity, suspended sediment concentration. We take samples from the pipe wall of the biofilm and the sediment, as well as various other measurements, to understand the mechanics of mobilization when it does occur. Now, one of the things that is really key about this facility is this pipe coupon system. So we've actually drilled holes in the pipe, and we've created these pipe inserts with 3D printing that fit directly into the pipe, and they allow the biofilm to basically grow on the inside of the pipe here. So we can take that out and put it under a microscope, or perform DNA sequencing of that material to actually get an idea of what types of bacteria actually exist there under different conditions. So this actually shows you here, this pipe coupon system here. So this is how the pipe actually fits, the coupon, actually fits onto the pipe.
Oh, I see that picture there. There's like 20 in that picture there. So how many are these just set up like every certain interval, or what's kind of like the background behind that? Because that seems like there's,
Yeah.
20 in a short amount of space. Are they kind of distributed amongst the whole facility,
Well, yeah, so like each rig has 11 rungs or 11
okay.
loops. And
Yep.
so we have 3 of those loops. One of them is located at the bottom, one in the middle, and one at the top. We call them monitoring segments
Mm-hmm.
that have a series of coupons on them. So it
Mm,
allows us to look at, and the coupons are in different circumferential positions. So we have coupons on the
mm,
bottom to
mm,
see what's going on the bottom, one on the sides, and one on the top. So we can look
mm.
at how the particles in the biofilm actually grow and are deposited on what position,
Yeah.
circumferential position. But we can also look at how they move or how they grow spatially or longitudinally. And that's why we have a bottom, middle, and top row.
Oh. Mm-hmm.
So when we have these coupons and they have, let's say, bacteria on them, we have microscopy facilities here that look at, basically, that can identify where the bacteria are and where these macro molecules are, like these proteins and lipids and polysaccharides that form the biofilm to protect them. We do this we can do this in a number of ways, but at Queen's here, we do it with fluorescence microscopy. So this shows you, this is a kind of a plan view of a coupon, and this is a 3-dimensional profile of that same sample. So what we do is, once we take the coupon out of the pipe, we stain it with different stains. So some stains adhere to the bacteria, other stains will adhere to certain macromolecules like proteins. And what the microscope does is that it actually it sends beams of light at different wavelengths, and the stains will be excited in a certain range of wavelengths and then re-emit at another known range of wavelengths. And so, of course, the camera will actually capture that. So what the wavelengths, the green range of visible light are the bacteria. And you could see where my cursor is, this line would be the wall of the pipe, so that you could see the bacteria close to the wall. And this is a macro protein or macromolecule that would form that biofilm. So you could see that it's on top of the cell. So it's actually, acting as a shelter, protecting those cells against all of the chaos that's inside pipe. Because the inside of a drinking water pipe is an extraordinarily hostile environment. Not only do you have shear force trying to rip you off the wall, you also have disinfectants that are trying to kill you. And to add injury to insult, there's nothing to eat. It's an extremely oligotrophic environment. In other words, very few nutrients. So these macromolecules actually serve a number of functions. Like I said before, they act as cement to provide structural strength to the biofilm so the bacteria don't get ripped off the wall. They also provide shelter against chlorine, so it makes it very difficult for disinfectant to diffuse into the biofilm and kill the bacteria. But at the same time, you have these little microchannels that allow whatever nutrients there are in the water above to diffuse into the biofilm itself, so the bacteria can eat. So bacteria are very, very versatile and adaptable, and in my opinion, very clever. They always find ways to survive despite, adversity. So that's kind of the facilities that we have to look at direct measurements of what's going on at the wall. And most of the action is at the wall. Now, we also have what's called bright field microscopy. So this is just an optical microscope with light that takes photos of the iron particles only. So we don't see any of the biofilm here because we're not using fluorescence microscopy. So after dosing the rig with these iron particles, what we did is we took coupon samples and we basically imaged them and basically looked at where the iron particles were. And we all sort of created a number of metrics to allow us to quantify the level of our particles found on the wall. And what we came up with was, what is the total area occupied by the particles divided by the total area of the field of view? So this shows you, these are typical images of what these particles look like when they're on the wall. So they obviously stand out, very visible red against the PVC blue. But this little graph here also shows you that so the bars, the details aren't important, but basically the x-axis is the time, starting at time 0 up to the 30-day period where we're dosing with particles. And you could see that the iron particles, the percent area occupied by the particles. So, sorry, we dosed at this time 0. And as it was recirculating through the loop, more and more particles were being settled out or adhered to the wall. You could see that the percent of total field of view is actually going up with time. And then as we're starting to flush, you could see that, so this is flush number 1, 2, and 3. So we flushed with 3 flow rates or 3 shears. You're actually mobilizing, dislodging those particles because with each successive flush, you have fewer and fewer particles. We also looked at particle sizes and how they're influenced. So basically, what happens is each of these plots are time plots. So this is at time 0, 7 days, 14 days. So every week, we looked at the particle size distribution of these particles on the pipe wall. And you could see that initially the big particles settled first, but then the smaller particles started to settle. So there's more a uniform distribution of particle size. And then as we started flushing, it's the small particles actually that were the large particles, that were preferentially mobilized, and the small particles were actually stayed in place. And the reason for that is, so this is sort of shows you a microscopic view of where the particles are on the pipe wall. So if you have this is the PVC wall that has some roughness. So to the naked eye, it looks smooth, but on a microscopic level, it has these little valleys and hills where the small particles can become lodged. And so the big particles can also become lodged, but they're not as firmly secured to the wall. So when
Mm,
the big flows come along when we flush, these large particles actually are the first ones to mobilize. And these little ones that are protected by the roughness of the pipe actually
mhm,
stay there,
mm,
which is counterintuitive because most people
yeah,
think that it's the larger particles that will stay on the wall and the smaller ones will be dislodged. So that was an
yeah.
interesting finding.
That's what I would have thought as well. So
Yeah.
just
So we did a number of things. We've been doing a number of things with this research. We sort of developed this unique PVC pipe coupon system that allows us to look at what's going on the wall. We looked at, how the size of the particle affects its sort of potential for mobilization, where the small ones tend to remain on the pipe wall and the large ones tend to be mobilized. We also looked at a lot of the factors that contribute to how many particles will adhere to the pipe wall. And a lot of our research now is moving in the direction of looking more at, the conditions that foster biofilm growth and
mhm,
how factors like temperature and, hydraulic shear force and things like that affect not only the physical properties of the biofilm, but also the microbiological ones. So, are there certain conditions that will favor certain types of bacterial groups over others? So those are all interesting things that municipalities care about because the overarching fear of municipalities is that, biofilms can become homes for pathogenic bacteria,
mhm,
which, if they become dislodged because of an event in the system, can
mhm.
cause a lot of people to become sick. So our research has been funded by a few agencies, primarily CFI, which allowed us to build this drinking water distribution lab, and NSERC, which is supporting most of our research program and our students.
Well, thank you. Yeah, that was a great presentation. Very much anything. Thank you so much. That's really, when I was looking over the slides at first and looking over kind of the project, I obviously knew that it was a large project, but when you say that it was a large project and you had to look at accumulation, that means that this project spanned even how long years? You said 4 years, 2018 to 2021? Is that what it
Well,
was?
we've been using the facility
For
since
7 years.
2017, so for
Wow.
7 years now.
Wow, wow. And was
Yeah,
this project, inspired by your work in consulting? Was, red water problems and such kind of such a large issue that it kind of brought you back into research? Were you doing research the whole time you were consulting? What kind of brought this project to life?
yeah. I mean, municipalities are always struggling with water quality.
Of
That's
course.
one of their day-to-day concerns, to make sure that they're delivering good water to customers. So it's always been an interest that I've had,
Mhm,
and a number of research questions that have very practical implications for municipalities. So when there was an opportunity to apply for a large grant to establish this facility, I thought it would be a good idea. It would serve the industry, the water industry in Canada, quite well. And so we're partnering with different municipalities like the city of Calgary,
mm,
looking at their water quality issues, as well as the city of Waterloo,
mm,
who have had some discoloration problems start of COVID, because they
mm,
saw big changes in the water demand patterns because of COVID, as
mhm.
people no longer going to work and staying at home
Yeah,
caused a lot of stagnation in parts of their system and started causing discoloration problems. So since partnered with us, and we're going to be starting a field investigation to try to get to the root causes of those discoloration problems. Yeah.
interesting. And so, even just a question related to some of the outcomes, for example, the particles and the PVC pipes, do you think that this work will kind of help develop new, even PVC design? Like, what would you hope? Would you just hope that you basically figured out kind of what factors, for example, foster biofilm growth or what conditions, encourage the iron particles to stick to the PVC pipes? Is
Yeah.
this going to be kind of redesigning? Are we looking into, but what is going to be kind of, you're hoping, what do you think
Yeah.
this will?
Well, yeah,
I know it's a difficult task to obviously.
no, no, no. We're trying to first understand the sort of basic science of particle deposition and adherence and particle mobilization. Once we have that understanding, then we can start sort of developing or adapting technologies to deal with it.
Mhm.
And so one of the things that we're starting to look at is a new flushing technique, for example.
Yeah.
And so we've started examining that with one of our students. And so conventionally what they do is they what they do unidirectional flushing. So they flush in one direction,
Mm,
usually in the same direction as the sort of normal everyday flows,
okay.
to try to mobilize the material that's adhered to the wall. So we're actually trying to see if flushing in the opposite direction to the everyday flow actually has an enhanced effect. We're calling it reverse unidirectional flushing.
Mm.
And the idea was sort of born from basically that little diagram that I showed you.
I was gonna say, is it how like the pipes are kind of like they kind of over
Yeah.
time they almost change shape based on water going on one side, almost like the little cracks and such?
Well, no, it was born from this idea here that the particles will, given the normal flow direction and flow
Mhm.
velocity, they will kind of find their natural habitat in the little,
Mm.
nooks and crannies of the pipe wall.
Yeah.
But, and if you flush in the same direction as the normal flow, you'll get some particles. You'll be able to mobilize some particles, but maybe not the ones that are nicely sheltered. But if you flush in the
Mm.
opposite direction, perhaps those particles will, because they're perfectly adapted to the forward direction, let's call it. Once
Mhm.
you reverse that direction, maybe they're actually going to be they'll be able to be mobilized more readily.
Mm.
So we're examining that too as a possible way of cleaning pipes.
Mm.
Another thing that we're doing with Dr. Jin, who's an expert in nanobubbles. So
Mm.
these nano-sized bubbles that we can introduce in the water of the pipe can also carry molecular disinfectants like
Yeah.
chlorine or chlorine
Mhm.
dioxide, things like that. And these little nanobubbles can diffuse into the biofilm. And then basically,
Wow.
like a Trojan horse, release their disinfectants and kill the
Mhm.
bacteria, right?
Wow.
So in trying to understand how biofilms work and how they form these little microchannels, then we can actually tailor a technology like nanobubble technology to get rid of biofilms.
Mhm. Wow, that's an interesting collaboration as well then.
Yeah.
Wow, very interesting. I mean, another question I have is kind of, do you see, I mean, obviously antimicrobial pathogens are obviously like almost like a silent pandemic right now going on. I myself worked
Yeah.
in an antibiotic resistance lab in university, so I'm really, interested in the topic. Besides kind of antimicrobial resistance, do you see any other kind of like up and coming almost trends that are going on with water quality besides red water that you can foresee kind of having an impact on this project? Right now, you said there's iron and manganese you're looking at, and you're looking at almost biofilms. Do you foresee anything else that you might be investigating in the future? Are there any new metals or anything going into water quality? What do you
Mhm.
kind of foresee with this?
Well, so we have two PhD students who are looking at how trace concentrations of antibiotics
Yep.
and metals like cadmium in the
Mm,
water, in drinking water pipes,
yep.
can basically promote, and you would know this because you worked in the area, can promote gene expression of the
Mhm.
bacteria that reside in the biofilm.
Yep.
Basically, mobilize these genes so that they can activate various resistance functions like an efflux pump function or other functions. And so what we're doing there is we have these bench scale PVC pipe loops, and we run water through them just like we do in the big facility. And then we dose them with trace levels, like sub-inhibitory levels of antibiotics and metals. I'm going to say about antibiotics first, over 30 to 60 days. And we basically take samples, RNA and DNA samples. We run qPCR
Mhm.
analysis on them and RT-qPCR to
Mhm.
look at whether these resistant genes are being expressed and used
Mm,
to activate different resistance mechanism. These are the first students in this area, and there's really hasn't been very much work done on the drinking water side of things.
mhm.
Most studies have just sort of taken samples from real systems and run qPCR analysis on them for a number of known resistance genes and said, "Yep, we found some genes. Don't
Yep.
know why they're being produced, or they're
Mm,
just." So we're trying to go one step further and try to look at, what factors can lead to the occurrence of these genes and their expression.
mm.
Now,
Wow.
on the wastewater side, there's a lot. This is a very active, as you may know, as a very
Very
active
active area.
yeah,
Yeah.
because the density of cells, of bacterial cells, is much higher, obviously, in
Sure.
wastewater than drinking water.
Yeah.
My initial impetus in starting this area of research on the drinking water side was that, these biofilms are extremely bacteria dense.
Yep.
So you have these bacteria that are, squeezed in cheek by jowl in these biofilms. And so that is, well, I guess the question is that a conducive mechanism for horizontal gene transfer, right?
Yep.
As opposed to just having cells floating around planktonically in the bulk water, not really interacting with their brothers and sisters. So we're really
Most
trying to
definitely.
look at the role of biofilm in promoting resistance in drinking water systems.
Yeah. Thank you so much for coming in, and we really look forward to seeing the future of your project and everything. Thank you.